Hi, this is Alex from devstories.eu. Scanpy, Single Cell Analysis, Episode 14 of 16. Just because two cell clusters sit next to each other on a UMAP plot does not mean they are biologically related. Visual proximity in two dimensions is often a mathematical illusion, and if you rely on it to draw developmental trajectories, you might connect dots that do not actually touch in high-dimensional space. To map actual, statistically backed connectivity, you need abstracted graphs with PAGA. PAGA stands for Partition-based Graph Abstraction. We need to clear up a common misconception immediately. PAGA is not a dimensionality reduction embedding like tsni or UMAP. It does not calculate coordinates for individual cells to draw a scatter plot. Instead, PAGA creates a simplified, coarse-grained graph. The nodes in this graph are whole clusters or partitions of cells. The edges connecting those nodes represent the statistical confidence that those clusters share a continuous boundary. When you call the PAGA function on your annotated data, you point it to a specific set of group labels, usually your Leiden or Louvain clusters. The algorithm then evaluates the boundaries between these groups by digging into the underlying single-cell neighborhood graph. It looks at the individual cells in cluster A and counts how many of their direct neighbors belong to cluster B. By tallying all these cross-cluster connections and comparing them to a random model, PAGA generates a quantifiable connectivity matrix. A high value means the clusters are deeply intertwined, suggesting a biological transition. A low value means they are separate islands. Consider a concrete scenario. You're tracking immune cell development, and you need to prove a specific progenitor cluster directly gives rise to an effector T-cell cluster. On a standard visual plot, the layout algorithm might throw an entirely unrelated cluster right between them, making their relationship look indirect. By examining the PAGA connectivity matrix, you bypass this visual distortion. You look directly at the mathematical edge weight between your progenitor and effector groups. PAGA allows you to set a minimum connectivity threshold. When you apply this threshold, you filter out spurious, low-confidence connections. If the strong edge between your two target clusters survives the cut, you have established a statistically robust link. This is the part that matters. PAGA doesn't just sit alongside your embeddings, it can fix them. Because PAGA preserves global topology so reliably, you can use the abstracted graph to initialize a UMAP embedding. Instead of letting UMAP start from a random spatial layout, you tell it to position the individual cells based on the coarse-grained PAGA graph. This anchors your final two-dimensional visualization to the true high-dimensional reality, ensuring that distant biological states are not artificially squished together. Visualizations will always distort complex data to fit it onto a flat screen, but the abstraction of a neighborhood graph relies entirely on mathematical proximity. Trust the graph over the picture. That is all for this one. Thanks for listening, and keep building.